And also, we will speak in English today, but 2 of our presentations will be in English by master students and 1 in Swedish. If there are questions, please ask in the chat, and every presentation should not take more than 10 minutes, because in case there are questions, we would like to have time for that. Please, Mohammed, the stage is yours.
All right. So my thesis is about monitoring the emissions of non-CO2 greenhouse gases from wastewater treatment plants. This is a case study that I was collaborating with MDU as a student there. If I start with, so if I start with why we monitor these greenhouse gas emissions from wastewater treatment plants, we see that methane and nitrous oxide has a higher global warming potential than CO2, where the numbers actually show that methane has 28 times higher methane than carbon dioxide. And if you compare it to nitrous oxide, the numbers show that it is up to 300 higher than CO2. And scientific reports show that wastewater treatment plants are a significant point sources of these gases. And we also see that non-CO2 gases are underreported when it comes to scientific reports showing emissions of those gases in WWTPs. So reports also show that there's a limited long-term measurement data available for WWTPs, and especially for Sweden, we see that methane and mostly nitrous oxide is not reported in the reportings, basically. So this study only focuses on Kungsängens WWTP here in Västerås. And this picture shows an aerial view of the field. Very hard to see in the picture, but there are some points that I color showed here in red and green and yellow. Very hard to see, but these are the points that I measured the gas concentrations in my study. I will show a better picture later on. So some of the measurement challenges included that we had an open air measurement, and this meant that it was sensitive to wind and rainfall. So for example, when there's a lot of wind, the measurement thing that we used would start going up and down, and there would be fluctuations in the reading data. And rainfall, it was sensitive to the actual analyzer that we were using, that we could not operate while raining. There are some challenges when it comes to spatial variability. We see that there are different zones generate different emissions, but in this study we only focused on one part of the biological treatment process. There are some temporal variability with fluctuations and seasonal changes, but when it comes to this study, it was only conducted during the summer months. So later on, and this is to mention, this is only the start of the project. So, but it is planned that it will be going on after the summer to see how seasonal changes also, if seasonal changes actually show something. So some of the challenges also include risk of missing emission peaks. The measurements that was conducted it would only go from 10 minutes to 30 minutes, and depending on rain and time availability. But to get the whole picture, it's important basically that longer measurement windows are conducted to see how peaks are performed. And we also see that there could be environmental interference, as I mentioned before. So there's also the operational change, the pole movement, and so on. And we see that currently there are no continuous plant level monitoring. Mälardalen is the company where we actually did this study on, and their measurements now only show that they report methane emissions, and this is measured once every 2 years, and it's done by EGMAT, and it doesn't show, they don't do measurements on nitrous oxide, which is very important point because nitrous oxide was shown to be a huge emission source in the WWTP. And it's also important to point out that those methane emissions were only looked at not in the biological treatment stage. So there's literally to no report that shows this. So this is our measurement approach, and the gas analyzer that we used is called GasMet, and it can analyze multiple gases. And for our study, we measured methane and nitrous oxide. And with it, you can see on the black screen right there, it's the tablet that we use to basically see the data and the inflatable pool. And to it, you can see that there is a tube going into the tube, basically, and also into the gas analyzer showing, and that's how the measurements were done. And the points that those measurements were done are the red point and green point and yellow point, which shows oxic tank inlet, return sludge line, and anoxic zone outlet. If we move on to the data collection and processing, so the measurement duration of it will be under 20 to 30 minutes per sampling point. And for the gas to work properly, it needed to be calibrated with nitrogen gas to flush it at MDU. And to deal with the data, CalcMet results data, Excel was used to produce graphs and so on. And also environmental factors were logged, so wind speed, temperature, to see how these play a role in different data. So this is one of the results, one of the day measurements, June 5th, and we see that for methane, that return sludge showed consistently a high methane concentrations, while the other two were kind of low. And if we compare to nitrous oxide on June 5th, we see that oxic inlet showed consistently high gas emissions, gas concentrations. So the concentration patterns show that highest methane concentrations was at the return sludge, and peak nitrous oxide occurred at oxic zones. Anoxic zone outlet consistently showed low on methane and nitrous oxide, and these results align with the previous findings that showed these results also. So to conclude, Methane and nitrous oxide concentrations, they vary significantly by treatment stage. So it's important to look all the treatment stages. And we see that Ristani sludge for methane and oxide zone inlet for nitrous oxide, those are major hotspots. When it comes to Ristani sludge line, it consistently showed lowest emissions. So recommendation and future work, measuring flow rate is important to actually quantify the total emissions. The study was only focusing on the concentrations of the gases, but to actually quantify the total emissions, measuring flow rates is important. And conducting continuous monitoring is also important to detect peak events and diurnal patterns. Future works also include exploring on-site mitigations options, for example, photocatalytic oxidation for both methane and nitrous oxide. And here are some of the key references that I used for my study. And this concludes my study. Thank you for listening.
Thank you, Mohammed. Great work. And if there are any questions, please don't be shy and ask. One very short question, Mohammed, since I'm not really, I have a bit of different background, but how about the values that you found, the concentrations of methane and nitrous oxide? What can we compare them with, these values? Is there any regulations, any guidelines?
So this study was only basically to first see how concentration, the gas concentrations for these two gases vary during this biological treatment process. And later on, when we actually know how much of the methane and nitrous oxide are actually emitted, then they could be compared to, for example, carbon dioxide, where we know that for methane, it's 28 to 29 times stronger than carbon dioxide, while nitrous oxide, it's up to 300 stronger. And this is measured over 100-year period.
Okay.
Yeah.
Good, interesting. Thank you. Please, Diane, prepare your presentation.
I can ask something for Mohammed.
Absolutely.
Thanks, Mohammed. Ulf Jeppsson here. So you didn't really measure the whole wastewater treatment plant. You measured in the activated sludge part.
Yeah.
I mean, in terms of methane, we normally have more from the anaerobic digesters and from the sludge deposit, the sludge storage part and so on. You had one sensor that you had to move around, or since you talked that it was fairly short measuring periods of N2O?
Exactly. So this gas analyzer, it simultaneously will measure basically for both methane and nitrous oxide. So when I conduct on one point, let's say the Ristani sludge, and then I would move the gas analyzer and the pool itself to another pool, and start new measurements there. And the duration of the measurement would last depending on how the data slows down and I see some kind of steady state. Then I would say, okay, then it will just repeat the same values here. But then there was some challenges with the pool itself. It would sometimes there would be a lot of wind, and it will start moving up and down, and this will show that the ambient air would come into the pool, and it would show some fluctuations in the readings, basically.
Yeah, and I think that there is a consensus within the community that maybe especially for N2O, it is important to have long-term measurements because it will vary very much depending on from week to week and from season to season of how much N2O is produced. You also I was surprised to see that it was selected that you would measure N2O and CH4 in the gas phase at the outlet of the anoxic zone. From if you look at literature, you would have seen that normally it's 0. If you measure it in the gas phase, if you measure it in the water phase, completely different. But in the gas phase, since you have no stripping, you have no oxygen coming in there will be very, very little showing in the gas phase.
Right. So these points were pre-selected by both my supervisor here, Sebastian, who have way better knowledge than I do, and also Mälardalen. And we could see the anoxic outlet zone. There is still some kind of oxygen levels still existing there, but not as much, of course, as the oxic inlet. But they have DO sensor that measures the oxygen level that exists in that area. But even if it's not as much as the oxic zone, the anoxic zone may sometimes also have some kind of oxygen there.
Yes, but you don't have active aeration, so you don't have the bubbles going through the water. No. And even if there's some oxygen in there, it will, unless the N2O is saturated in the water, it will stay in the water until it reaches the inlet of the aerobic zone, and then it will start to get stripped with the air bubbles.
Correct.
Thank you. If there is more questions or observations, please continue in the chat. Thanks again, Mohammed. And Dian, the stage is yours, and let's hear more about microplastics.
Okay, I hope you can see my screen.
Yes.
Perfect. All right. So my name is Diane, and I did just recently do my master thesis at KTH, and I will be presenting today about microplastics in sludge across Swedish wastewater treatment plants. So microplastics are considered as small plastic particles that range up to 5 millimeters, and they're found in form of fibers, flakes, and grains. They're classified into primary and secondary microplastics, and if you look at the cycle, most of the microplastic come from domestic behaviors such as toilet flushing, bathing, dishwashing, and doing laundries, but there is also industrial influence and also leachate from the roads. And so the main objective of this wastewater treatment plant is to clean this water and put it back to the ocean or fresh water, and to kind of close the water cycle. The sludge is a solid byproduct that is usually used in agricultural for fertilizing because it has a high content of nitrogen and phosphorus. Everyone is familiar with the steps in the wastewater treatment process. The sludge that we get from wastewater will be treated, including the external sludge, through mainly 3 processes and will be disposed in different ways. In my thesis, I focus on agricultural use. And here you can see the different sludge samples during different stages. And I will talk about how the microplastics land there, and yes. So my 3 main research questions is to analyze the physical-chemical characteristics of these microplastics found in the sludge samples, to find out the factors that influence the occurrence, and also how much of the sludge is being disposed in the environment. And these questions help to understand the fate of the microplastics in sewage sludge, but also to assess the amount of the disposal into the environment. I got 3 replicates, or I was working with the measurement results from 3 replicates that were received from 7 wastewater treatment plants around the Mälardalen region. And so these samples were then, the microplastics were then extracted from these sludge samples and then used in the FDIR measurements. And this gave out a result of a spectral fingerprint, which was then compared to the reference spectrum found in the simple software. And then my job was kind of to analyze these measurement results. And I made some calculations and analysis, and to complement my results from the analysis, I also did online interviews with the representatives from the wastewater treatment plants, and also sent them a questionnaire to find out the key figures of the plant. So here is a distribution of the population equivalent across all the 7 treatment plants. The plant with the highest population equivalent is usually the biggest one, or the one that can treat the most wastewater, and accordingly the amount of sludge matches the amount of wastewater that can be treated. So Shepala would have been is the largest plant that I was working with, or the measurement data. And here there is a summarization of all the factors that may influence the presence of microplastics in wastewater. The inlet mix, which contains municipality wastewater, industry inlet, and also hospitals, there might be also influenced by the external sludge that contains usually food waste, fat, and grease from restaurants, but also sludge from smaller wastewater treatment plants. Another factor could be the opening of the fine screens found in the primary treatment. And through the questionnaire, it was also confirmed that every plant that was part of the research uses polymers during wastewater treatment, but also polymers called polyacrylamide in the sludge treatment. Here there are some images from the samples to show how the average samples looked like. There was one sample that showed a bit more larger microplastic content and overlapping particles, and this was also then considered as the outlier and excluded from future comparative analysis between the plants. So now here there are only 6 plants. There is the distribution of all the particles, plastic particles found in number of particles per gram of total solids. The most occurring microplastics are polypropylene, polyethylene, and polyester, accounting altogether to almost 85% of the detected microplastics. And here there is the mass, so nanograms of microplastics per gram of total solids. Here the most occurring microplastics are again the same types of microplastics, but this time they account for almost 98% of all the detected microplastics. The count is not directly proportional to the mass. This might be influenced by the density, the type, and the shape of the microplastics. To find out the morphology, three values were calculated: the length-width ratio, the elongation, and the compactness value. And the trend showed that most of the plastics that were found are fibrous, and the areas were also categorized into 4 categories: very small, small, medium, and large sized. So the most of the particles found fall into the medium sized category. And yes, the last step was for me to calculate how much of these microplastics land in the agricultural soil through the sludge used for fertilization. So 5 out of the 7 wastewater treatment plants use it for agricultural purposes. And without including Nodra's results, 14,600 tons of sludge per year goes goes to the agricultural soil as fertilizer. And this makes up almost 11 tons of microplastic that also go annually into the soil. This accounts for 0.1% of the sludge amount produced yearly. And yes, the 2 other wastewater treatment plants that don't use it for agricultural purposes, they use it for construction soil, landfill, and even incineration. To answer my research questions, so the most common found microplastics in sludge are polypropylene, polyethylene, and polyester, which often are found in packaging, cosmetic use, and also in the clothes, synthetic clothes that we wear. The average count was about 1,300 microplastic per grams of total solid, compared to the other studies, for example, Germany and United Kingdom, who used similar extraction method, show different results. And this shows again how important it is to standardize methodologies for research like this. And also the fine screening in the beginning has shown to filter out larger particles of microplastics in the later stages. There has been a variability even between the samples gotten from each plant. This is because of the batch system and the temporal change. And it has also shown that larger wastewater treatment plants who treat larger volumes have a much better removal efficiency of the microplastics. The 4 factors that may influence the abundance are domestic behavior, which is the most major one, followed by industrial influence, external sludge, and the polymers that are used during the treatment process in the treatment plants. The types of disposal methods are landfilling, incineration, construction soil, and the most common one, agricultural fertilizing. And this is also a common practice because the European Union encourages to reuse the sludge as fertilizer. And from the results, it shows that almost 11 tons of microplastics land in the agricultural soil just from the 4 treatment plants, and makes it also reconsider certifications such as REVAC, for example, to include like a limit of microplastic load or microplastic content that should be found in the sludge. Once the microplastic land in the soil, they can be transported by wind, water, and even plowing from tractors, so through agriculture activities. And from the soil, it lands in the beverages and food and may cause health risk. Before I conclude, there are some things that each of us can do at home, or good practices that we can take with us. I'm not going to go much into details, But I would like to give a special thanks to all of the wastewater treatment representatives who got involved during my thesis, for Clara for doing the measurements, and also Professor Sahar for giving me the opportunity also to present here today. Thank you.
Thank you. Thank you, Diane. Thank you. Very, very interesting. Thanks a lot. Great work. Any direct questions from the audience? Very short, because we need to go on to the last presentation. Otherwise, please put your question in the chat. Diane, I would like to ask you just very, very short, because I have been working myself with microplastics in the stormwater context. And it's interesting to see that you are using FTIR, because it has been one of the first measurement or analysis techniques that have been used. And as we all know, this is not standardized, the microplastics analysis. And also just quick, how you came from number of particles to the mass of microplastics, because there are other techniques, as you know, that are actually quantifying already microgram per liter or microgram per gram sludge microplastics.
So in the measurement results that I got, so were already the number of particles that I could calculate from. And each particle that was discovered there was also the mass given. So I just scaled it up from the subsample to the grams of sludge or dry sludge. Yes, so the mass was per particle was already given.
Yeah. Okay, I see.
Yeah.
Okay, interesting. Thanks a lot. Great. Thanks, Diane. And Jenny, please, if you can put up your presentation.
Yes.
And Jenny, you will talk about the EFAT technique and the biological phosphorus and nitrogen treatment. And maybe, take it in Swedish as you wish.
Kan ni se min skärm nu?
Ja.
Ja, vad bra. Ja, mitt namn är då Jenny och mitt examensarbete har handlat om biologisk rening av kväve och fosfor vid låga temperaturer med IFAS-processen då. Och exjobbet är gjort på Mittsverige vatten och avfall i Sundsvall och är en del av ett forskningsprojekt som pågår där. Så först tänkte jag dra lite kort bakgrund till det forskningsprojektet. Och bakgrunden är att det har kommit nya krav från EU då som ställer högre krav på både kväverening och klimatneutralitet. Och samtidigt så är också den framtida tillgången på fällningskemikalier osäker. Och i norra Sverige så blir de här ökade reningskraven särskilt utmanande eftersom de bakterier som används för biologisk rening jobbar långsammare i kallare vatten. Och för att möta dessa ökade utmaningar då så krävs det nya tekniska lösningar. Och det är här som forskningsprojektet KallPN kommer in. Där Mittsverige vatten och avfall och IVL samarbetar för att ta fram resurseffektiva reningsprocesser med låg klimatpåverkan anpassade till kalla temperaturer. Så mitt bidrag i det här forskningsprojektet har varit att undersöka reningsprocessen i fas och se hur effektivt den reningsprocessen kan avskilja fosfor och kväve biologiskt vid låga vattentemperaturer mellan 6 och 10 grader. Och fokus har varit att se hur olika driftparametrar såsom kolkälledosering, slamålder och redox påverkar reningseffektiviteten. Samt hur temperaturen påverkar fosforsläpp, som alltså är det första steget i biologisk fosforrening. Och i en I-fas så passerar avloppsvattnet flera tankar där särskilda förhållanden råder för att gynna olika typer av bakterier som bryter ner näringsämnena och på så sätt renar vattnet. Och bakterierna finns då både i det aktiva slammet men också i biofilm på bärare. Och biofilmen gör bakterierna mer tåliga mot bland annat lägre temperaturer. Så I-fas är alltså en reningsteknik som kombinerar rening genom aktivt slam och på bärare. Och bara lite kort bakgrund om de här biologiska reningsprocesserna. Biologisk fosforrening sker i 2 steg. Först så sker ett fosforsläpp under syrefria förhållanden där bakterier som kallas för PAU tar upp kolkälla i form av VFA och släpper ut fosfat. Och sen i senare steg så sker ett fosforupptag under syrerika förhållanden där fosfat tas upp igen, vilket gör att nettofosfor kan avlägsnas från vattnet. Och biologisk kväverening sker genom nitrifikation och denitrifikation. Och nitrifikationen kräver syre medan denitrifikationen kräver kolkälla och sker utan syre men med nitrat. Ja, så studien genomfördes i en IFAs pilotanläggning intill Fillans avloppsreningsverk i Sundsvall. Och vattnet till piloten togs från försedimenteringen inne på verket då. Och piloten bestod av 7 olika tankar med olika miljöer för att gynna både kväve och fosforrening. Och under perioden jag kollade på, från januari till april ungefär, så ändrade vi olika driftparametrar för att se hur det påverkade reningen. Och vi ändrade parametrar dels för att optimera processen men också för att testa prestandan av den. Så vi ändrade bland annat temperaturen, fyllnadsgraden av bärare i de olika tankarna, kolkälledosering och slamålder. Och för att följa effekten av de här ändringarna vi gjorde då, så använde vi både online-sensorer som fanns i tankarna och vi tog också manuella vattenprov som vi analyserade 1 till 2 gånger i veckan. Ja. Och resultatet då visade att nitrifikationen fungerade väldigt bra under hela perioden och ammoniumreduktionen låg stabilt runt 99% oavsett förändringar. Så det visar att IFAS kan ge en väldigt stabil nitrifikation även vid låga temperaturer. Och när det gäller kväverening så varierade denitrifikationen mest. Och framförallt så såg vi då att tillgången på kolkälla var väldigt avgörande. För när kolkälla doserades så fungerade denitrifikationen bra. Men när doseringen stoppades så ökade utgående halter av nitrat från piloten. Och det visar då tydligt på att kolkälla krävs för effektiv denitrifikation i kalla temperaturer. Och fosforreningen förbättrades successivt under hela perioden. Och vi kunde se att fosforsläpp i början av linjen var beroende av helt syrefri miljö. För när redoxpotentialen ökade, till exempel på grund av att nitrat kom genom returslammet då, så försämrades det här fosforsläppet. Så det är alltså viktigt att minimera syretillförsel och nitrathalter i de anaeroba miljöerna för att gynna fosforsläpp. Och slamåldern visade sig också påverka. För att i början så tog vi inte ut något överskottslam, vilket ledde till en högre slamålder då. Och då försämrades också, eller fosforreningen var sämre i början. Och det berodde då troligen på att slamåldern var högre och att slammet då blev mineraliserat. För att när vi tog ut överskottsslam så förbättrades också reningen av fosfor. Ja, och sen för att studera hur temperaturen påverkar fosforsläppet då så genomförde jag även separata laboratorietester där slam från piloten utsattes för syrefria förhållanden och VFA vid olika temperaturer. Och resultatet visade att fosforsläppet gick långsammare vid lägre temperaturer. Och jag beräknade också en temperaturkoefficient på 1,06. Och det innebär i princip att reaktionshastigheten ökar med 6% för varje grad temperaturen ökar inom intervallet 4 till 8 grader. Ja, så slutsatsen av mitt exjobb visar då på att det är möjligt att uppnå en effektiv biologisk rening av både fosfor och kväve med IFAS under låga temperaturer. Men det också krävs det att vissa driftparametrar optimeras eller justeras, såsom kolkälledoseringen till fosforsläppet och denitrifikationen, slamåldern och nitrathalter. Och utifrån fosforsläppsförsöken går det också att se att reaktionshastigheten blir långsammare vid lägre temperaturer. Ja, tack.
Tack Jenny, tack för en snabb och koncis presentation. Tack för att du har hela tiden tagit upp hastighet